Hello, everyone. My name is Anurag, and I will be presenting the current evidence for serotonergic dysfunction in Parkinson's disease. Parkinson's disease is a neurodegenerative disorder which characteristically leads to alpha-synuclein deposition and damage within the pars compacta component of the substantia nigra. Although this disorder presents primarily with tremor, rigidity, and bradykinesia, the presentation is variable and can include other motor and a large range of non-motor symptoms. Serotonin is a neurotransmitter in the brain and other regions, such as the gut. Its chemical name is 5-hydroxytryptamine. It can have a range of behavioral and other CNS effects. It has known involvement in the psychopathology of many psychiatric conditions. Serotonin acts via 5-HT receptors. There are 7 families of this, of either G protein or ion-gated type. The diagram on the right summarizes serotonin synthesis, release, and action in relation to a neuronal body. Once serotonin is present in the synaptic cleft, its action can be terminated by either neuronal uptake through SERT receptors or enzymatic degradation. This diagram from a review article by Vega Suarez et al. 2019 depicts some of the key serotonergic modifications based on human and animal studies. In the control setting shown on the left, all components are healthy. Then the Parkinsonian serotonergic neuron shows alpha-synuclein accumulating and leading to mitochondrial dysfunction, reduced serotonin release, and death of the serotonergic neuron. The right side of the diagram depicts a special case called levodopa-induced dyskinesia, which I will discuss later. The dorsal raphe nuclei are a cluster of nuclei found within the brainstem in the brain and are the largest repository of serotonergic neurons. They send projections to almost all brain areas, including the basal ganglia. In Parkinson's disease, it has been noted that this area undergoes dystrophic changes. While dopamine is largely responsible for the characteristic motor features of Parkinson's, the serotonergic system has been shown to have a large influence on non-motor symptoms. One such condition is depression. An umbrella review of serotonin in major depression has concluded that the serotonin theory of depression, which has for years formed the neurochemical basis for depression, is not empirically substantiated. It reports that there is no consistent evidence for the hypothesis that depression is caused by lowered serotonin activity or concentrations. However, SSRIs have indeed been proven to be effective in treating the symptoms and preventing relapses of depression. More specifically, in Parkinson's disease, SSRIs improve depressive symptoms and freezing of gait. Studies in Parkinson's disease have shown reduced concentrations of plasma serotonin and its metabolite 5-hydroxyindolacetic acid, which also correlate with the severity of depression and pain. A postmortem study has confirmed lower levels of serotonin and its metabolites within the striatum of Parkinson's disease patients. Apathy is another non-motor symptom in Parkinson's disease. A PET study comparing apathetic and non-apathetic Parkinson's disease patients found that non-apathetic Parkinsonian patients mainly showed dopaminergic denervation and preserved serotonergic innervation. Meanwhile, apathetic Parkinsonian patients exhibited dopaminergic and serotonergic degeneration. Part D on the lower right here maps red and yellow colors along the right caudate nucleus, meaning higher 11C DASP binding in that region in non-apathetic patients, indicating lower SERT binding in the apathetic group. There is also evidence of the impact of serotonergic dysfunction in Parkinson's disease on non-motor phenomena such as sleep, fatigue, and interest. Insomnia and rapid eye movement sleep behavior disorder are common in Parkinson's disease. A study has shown reduced SERT density in Parkinson's disease patients with sleep disturbances in the striatum, raphe, and hypothalamus. Parkinson's patients tend to be fatigued. This is associated with reduced SERT binding density in limbic areas, and its severity inversely correlates with serotonin levels in the CSF. Another study related serotonergic dysfunction to hallucinations. There is also some impact of serotonergic dysfunction on motor symptoms. One such symptom is tremor. It has been thought that tremors do not arise due to dopamine alone. One such clue for this is the poor response of a Parkinson's resting tremor to dopaminergic medications. In addition, PET studies have failed to show a correlation between tremor scores and dopamine terminal capacity or D2 receptor availability. On the other hand, low doses of clozapine a drug with antagonistic effects to some serotonin receptor subtypes, has shown improvement in tremors in Parkinson's disease. Chronic SSRI use can indeed also induce tardive dyskinesia or Parkinsonism. Another motor symptom impacted by serotonergic dysfunction in Parkinson's is levodopa-induced dyskinesias. This involves the generation of abnormal movements in patients who are chronically managing their symptoms with levodopa. As seen on the right, levodopa-derived dopamine competes with serotonin for entering exocytic vesicles. The accumulation of dopamine and serotonin in the cytoplasm induces the synthesis of toxic compounds harmful to the cell. This is proposed to be a mechanism for these abnormal movements, although it remains poorly understood. Some literature suggests that levodopa-induced dyskinesias are due to serotonergic neuronal hyperinnervation, although there is limited and heterogeneous proof of this. A systematic review of PET studies illustrated a higher serotonin to dopamine ratio in dyskinetic Parkinson's patients, but suggested that dyskinesia may have a multisystem origin. The only drug currently available on the market which reduces levodopa-induced dyskinesias is amantadine, an NMDA antagonist. Dopamine and serotonin are believed to have some linkage due to concurrent pathologies in their systems. However, the verdict is still out on how serotonergic transmission modulates dopaminergic activity. In terms of understanding the timing of dysfunction, a study was carried out on carriers of the autosomal dominant A53T point mutation in the alpha-synuclein gene. This mutation confirms a highly penetrant risk of developing Parkinson's disease, which is almost indistinguishable from idiopathic Parkinson's disease. Using PET scanning, this study determined that serotonergic pathology occurs in the premotor stage in A53T SNCA carriers. It also precedes the development of dopaminergic pathology and motor symptoms. Another study showed that Parkinson's disease patients receiving dopamine-rich fetal grafts implanted in their brains show improvement in motor symptoms, but still suffer from non-motor symptoms. Functional MRI in these patients shows restoration of dopaminergic connectivity, but PET scanning shows reduction in 5-HT uptake binding. This is a scatter plot from the same study. It shows reductions in 11C DAS binding values in individual raphe nuclei for 12 Parkinson's patients compared to 12 normal controls. Dopamine graft transplanted Parkinson's patients 7, 13, and 15 had even more marked serotonergic dysfunction. In conclusion, serotonergic dysfunction is present in Parkinson's disease and affects non-motor symptoms and specific motor symptoms. Limitations include the replicability of studies, individual differences in participants, and complicated, poorly understood mechanisms, given that there are concurrent region-specific interactional variable effects of serotonin depending on location and receptor subtype. Studying serotonin in Parkinson's disease remains important, however, as this will aid in understanding the mechanisms underlying Parkinson's, helping in discovering novel biomarkers and maybe a potential target for pharmacotherapy in the future. Thank you for listening. My references are attached with this QR code.